Good morning, students. We are going to start with the lesson number 1, reproduction in lower and higher plants. We have studied in the earlier standards about reproduction. It is a process or it is a production of young ones like parents. We have also studied that reproduction is of two types, asexual reproduction and sexual reproduction. There are various types of asexual reproduction in which only single parent is involved, that is, no union of male and female gamete takes place in the asexual reproduction. So, fragmentation, budding, spore formation, vegetative propagation like cutting, budding, grafting, and tissue culture, these are all types of asexual reproduction. Now, let us learn about sexual reproduction. Sexual reproduction is nothing but union of male and female gamete in which flower is a sexual part or sexual organ of the plant. It is specialized reproductive organ of plant. It produces haploid gametes. The process involves meiosis and fusion of gametes to form diploid zygote. It produces genetically dissimilar offsprings because you know that there is a variation takes place in the sexual reproduction as male and female gametes are united. So half of the genes of male and half of the genes of female are combined, which gives rise to a new offspring. Sexual reproduction is characterized by fertilization, that is union of male and female gamete, formation of zygote. The result of fertilization is zygote, and then embryogenesis, which takes place in two stages, that is megasporogenesis and microsporogenesis. Megasporogenesis means formation of female gamete and microsporogenesis means formation of male gamete. We have studied about flower. It is a reproductive part of the plant. Some flowers are unisexual while some flowers are bisexual. Complete flower has 4 different whorls, that is calyx, corolla, androecium and gynoecium. Out of these 4 whorls, calyx and corolla are accessory whorls while androecium and gynoecium are essential whorls. The male reproductive organ is androecium, which is made up of anther and filament, and female reproductive organ is gynoecium, which is made up of ovary, style and stigma. You can see here in the nearby picture about the different parts of the androecium and gynoecium. So here you can see in the flower there is an anther. Anther is attached to the filament that gives rise to stamen, or it is an androecium. Then ovary, it is a part of the gynoecium. At the base of the gynoecium there is an ovary present. Then in anther you can see microsporogenesis take place, that is development of pollen grain. And in ovules you can see megasporogenesis take place, that is development of megaspore mother cell. Let us now learn about structure of anther. An immature stage of anther is represented by group of parenchymatous tissue surrounded by single layered epidermis. Anther is generally diathecous, that is, it has 2 lobes and tetrasporangiate. Each monothecous anther contains 2 pollen sacs. So, in diathecous anther there are 4 pollen sacs present, and therefore it is called as tetrasporangiate. Let us now learn about TS of anther. Very important topic. Draw and describe may come for 4 marks. Draw and describe may come for 3 marks, or only diagram also may come for 2 marks. So please practice this properly. So transverse section of anther, it shows epidermis. It is a outer single layer tubular cells. This is a protective covering or protective layer of the anther. Then there is endothecium, radially elongated cells, fibrous cellulose, thickening, hygroscopic. Then there is a middle layer. It is also a protective layer. It disintegrates at maturity. That means when anther becomes matured, this layer disintegrates. Then tapetum, It is a nutritional layer or food layer. Then pollen exine formation. It is required for the pollen grain exine formation. We have studied in the practical about the exine and intine of the pollen grain. The outer part is called as exine, inner part or inner layer of the pollen grain is called as intine. So formation of exine of pollen grain, it takes place by tapetum. Then microspore mother cell, meiosis and formation of pollen grains. Let us now discuss about microsporogenesis. Each microspore mother cell divides meiotically, that is here meiosis takes place to form tetrad, that is pollen grains. Here is a structure of pollen grain. You can just go through the structure of microspore or pollen grain. You can see the outer layer is called as exine, while the inner layer is called as intine. Pollen grain is non-motile and haploid in nature. It is surrounded by two layers, that is sporoderm. It has two layers, exine. The outer layer exine is thick and it is made up of complex non-biodegradable substance called sporopollenyl. It is smooth. Germ pore facilitate pollen tube germination. Intine, inner layer, it is thin, elastic and it is made up of cellulose and pectin. It forms pollen tube. In stage E, you can see two male gametes are formed from the generative cell. And in stage F, intine, it is giving rise to development of pollen tube. In this pollen tube, two male gametes they enter. Let us now study about structure of anatropous ovule. Each ovule develops inside the ovary and is attached to the placenta by a small stalk called funiculus. So you can see here funicle or funiculus. The place of attachment of funiculus with the main body of ovule is called hilum. In angiosperms, the most common type of ovule is anatropous, in which micropyle is directed downward and is present adjacent to the funiculus or in the funicle. So here you can see in this picture the stalk and the body. These are the two parts of the anatropous ovule. Stalk is called funicle. Body shows various parts like nucellus. It consists of central parenchymatous tissues. Then integuments. These are two protective covering. Then micropyle. It is a narrow opening at the apex of cell. Then chalaza, at the base of ovule present opposite to micropyle. And then embryo, female gametophyte, oval multicellular structure embedded in nucellus. Megasporogenesis, it is a process of formation of haploid megaspores from diploid megaspore mother cell. Now from diploid to haploid, that means this division is a meiotic or meiosis. Megaspore mother cell becomes distinguished in nucellus, more or less in the center but towards micropylar end of ovule. Here are the stages of development of female gametophyte. Megaspore mother cell, it gives rise to 4 megaspores which are haploid. Out of these 4 megaspores, 3 degenerates and 1 remains functional. It undergoes 3 successive free nuclear meiotic divisions. And at the end, total 8 nuclei are formed, 4 of which are located at each pole. So 4 and 4, total 8 nuclei are formed in the development of female gametophyte. You can see here embryo sac. At the chalazal end, there are 3 antipodals. At the center, central cell, it has 2 polar nuclei. Then at the micropylar end, where micropyle is present in the anthropous ovule, you can see there is a egg cell and 2 synergids. So total 8 nuclei are present, out of which 3 antipodals, 1 is egg cell, 2 synergids and 2 polar nuclei. I hope you must have understood about the formation of microsporogenesis and megasporogenesis. Now we are going to study about pollination. We are aware about the definition of pollination, that is transfer of pollen grain from anther to stigma. Transfer of pollen grain from anther to stigma. We have studied in the practical about the pollen grain, that we have studied 2 types, that is one is insect pollination and one is wind pollination. But there are 2 major types of pollination, that is self pollination and cross pollination. In self pollination, transfer of pollen grains take place within same plant, while in cross pollination, transfer of pollen grains with other plant or another plant. Self pollination, it is of 2 types, autogamy and geitonogamy. In autogamy, bisexual flower is pollinated by its own pollen grains. That means transfer of pollen grains of the same flower in the takes place on the stigma of the same flower. Then in geitonogamy, pollen grains are transferred from anther to stigma of another flower produced on the same plant but different flower. While in cross pollination, there are 2 types, like xenogamy, transfer of pollen grains from one flower to another flower produced on the different plant but same species. And here in the hybridization, there are 2 different species. That means pollen grain transferred from anther to stigma of the different species or different plants of the different species also. So there are total 4 types of pollination, 2 from the self pollination and 2 from the cross pollination. Self pollination. Pollen grains are transferred from anther to stigma of same flower or different flower produced on the same plant. Autogamy and geitonogamy, these are the 2 types of self pollination. Now in autogamy, bisexual flower is pollinated by its own pollen grain. Offsprings are genetically identical to their parents, for example pea plant. Geitonogamy, pollen grains are transferred to the stigma of different flower on the same plant. Do not bring genetic variation but ecologically significance, like cucurbita. Here are some advantages of self-pollination. Sure method of pollination has no role of external agent. Economical for plant, as no modifications like attractive flower, nectar, fragrance required. Least wastage of pollen grains. Pollen grains are not at all get waste. Maintenance of genetic stability or pure line in progeny. Here are some disadvantages of self-pollination, like no chance of forming improved varieties like hybrids. So hybridization is not possible here in the self-pollination. No elimination or introduction of characters in offspring is possible. Continuous self-pollination leads to inbreeding depression. Then progenies become weaker, disease susceptible, less productive and less adaptive. Does not favor variation and evolution. That means no variation take place, no evolution, because it is a self-pollination. In cross-pollination, transfer of pollen grains from anther of one flower to the stigma of another flower produced on the different plant or dissimilar genetic makeup or dissimilar genetic species. So already we have discussed about 2 species, that is xenogamy and hybridization. Now in xenogamy, Pollen grains are transferred to the stigma of different flower belonging to same species. Offsprings are genetically different. For example, papaya. Papaya is a unisexual plant. Then hybridization. Transfer of pollen grains from anther of one flower to the stigma of another flower produced on the different plant of different species, different varieties. That is, it gives rise to hybridization. So pollination between two species of varieties of cotton, that is a very good example of the hybridization. In market also we get different varieties of fruits of different nowadays we are getting hybrid oranges, hybrid guavas. So those hybrid plants are formed by this cross pollination. Here are some advantages of cross pollination. It includes genetic recombinations and brings variation. So here we can see variations which cannot be seen in the self pollination. Then characters can be eliminated or introduce new characters in the offsprings. Then production of improved varieties like hybrids by hybridization. Then progenies are strong and disease resistant, adaptive to environment and show better vigor. Then favors process of evolution. Disadvantages of cross pollination depends on external carriers, so failure chances are more. Not economical, as lot of energy is wasted for attracting pollinators or agencies. Wastage of pollen grains. Chance of elimination of desirable characters or wanted characters or introduction of undesirable characters. The genetic purity of offspring is not maintained. Agencies of pollination or agents of pollination. Here are some agents which help the process of pollination. Pollinating agents. There are some biotic and some abiotic agents. Abiotic agents are wind, water, and biotic agents are insect, bat, birds. In abiotic agent, wind, water. In water, hypohydrophily and epihydrophily. Let us now study about pollination by wind. Crop plants are wind pollinated. For example, wheat, rice, maize, grass, palm, etc. Floral adaptations in anemophilous flower. What is meaning of anemophilous flower? When pollination takes place by wind, that particular flower is called as anemophilous flower. Flowers are small, colorless, without nectar and fragrance. Pollen grains are light, dry, and produce large in number. Pollination by water, found only in 30 genera of aquatic monocots. For example, Vallisneria zostera. Some floral adaptations of hydrophilous flower. Hydro is nothing but water, and hydrophilous flower means the pollination which takes place by water. Flowers are small and inconspicuous. Perianth and other floral parts are unwettable. Pollen grains are long and unwettable due to presence of mucilage. Nectar and fragrance are absent. So nectar, nectary gland and fragrance are absent, so they cannot attract insect for the pollination. Hydrophily is of two types. Hypohydrophily, that is pollination occurs below the surface of water. Pollen grains are heavier than water, sinks down and are caught by female flowers. For example, zostera. Epihydrophily, pollination occurs above the surface of water. Pollen grains are light and floats on the water surface. Specific gravity of pollen grain is equal to water and reach the stigma of female flower. For example, Vallisneria. Insect pollination, it is called as entomophily. For example, rose, jasmine, then hibiscus. Adaptations in entomophilous flower. Flowers are large and showy, usually brightly colored. Have fragrance and nectary glands. Stigma is hairy and sticky. Pollen grains are spiny and have pollen kit. Special mechanism in some plants like salvia, lever mechanism to help pollination. Pollination by birds, it is called as ornithophilous plant. The plants which show pollination by birds are called as ornithophilous plant, and that pollination is called as ornithophily. So for example, bottle brush, butea, etc. Adaptations in ornithophilous flowers. Flowers are large, showy and brightly colored. Secrete abundant dilute nectar. Pollen grains are spiny and sticky. Flowers are without fragrance. Corolla is thick, fleshy, tubular or funnel shaped. Pollination by bats. Anthocephalus and Kygelia. These are the examples. Pollination by bat is called as chiropterophily. Adaptation in chiropterophilous flower. Flowers are with dull color with strong odor. Flowers are large and stout. Flowers open during night. That means bat is a nocturnal animal and therefore these flowers also open during night. Secrete abundant nectar. Flowers produce large amount of edible pollen grains.